Morning, everyone. Just let's, start in 8:03, like in 3 minutes or so, just letting people in. Okay. Good morning, everyone. First of all, I want to say first, like, how can I put it? Uh, I'm sorry for if I'm gonna be a bit slow today. I've not had a lot of sleep. They hit Aisha. I live in Verdun, that's literally what 1 minute away from my house. So I woke up in the middle of the night at like or 5 in the morning, and I've not been able to sleep back then. So I'll be a bit slow today, but bear with me. So I'm gonna share my screen. Right. Stop the video for bandwidth. Okay. Okay, guys. So last time, so I'm gonna go back here. So I really like this picture here. Yes. So here we have basically your glucose molecule, right? So this is your glucose molecule that we're trying to, let's say, extract as much energy. What we're trying to do in per se is we're trying to rearrange all the electrons, all the bonds to break them and basically give us, ATP at the end of the day. And why we do that? We do this basically glycolysis. We do this in the cytoplasm. We rearrange it, we break it down into pyruvate, into basically, so this is a six carbon into basically two or three carbons. And then this pyrite, the fate of it, it all depends on whether there's what happen if is there oxygen or not. If there's oxygen, great. We can further oxidize this and basically keep getting more energy. If not, this has to basically become the electron acceptor now because while we're basically breaking down glucose, we're producing a lot of NADH, a lot of these electron carriers. Because, as I said, the electrons moving around are dangerous and they basically lose just a lot of energy. So we basically have a look if there's basically oxygen. And if there is, great, this is being transported into your mitochondria. Then here using this massive complex, multi-enzyme complex called pyruvate dehydrogenase, it's going to literally oxidize this and to pyruvate base into basically acetyl-CoA. So you remove a CO2, and then this enters a citric cycle in where we actually start to further kind of oxidize acetyl-CoA. So we produce a lot of NADHs and also this FDH2, meanwhile, which is great. But now we have all of these electron carriers, this NADH, this FADH2, something has to happen. By this point, we need to basically, first of all, recycle them back into NAD plus or FAD plus, or sorry, FAD, because we there's a certain, let's say, amount of NAD that we have in our body. So we need to recycle them back. If not, we deplete our reserve of NAD. And that again is dangerous because now we don't have anything that's going to accept electrons. And electrons can now move freely all around. And this can really be a huge issue. So what happens? So now if we go all the way here. So now, as I said, we've produced a lot of NADH and FADH2s, basically during our whole breaking down of of a glucose molecule. So what we're trying to do right now is we're trying to pass on to remove these electrons. So these have to be basically oxidized. They need to go back. And they're actually transported into basically certain other complexes. And these complexes, well, we're going to be calling them right now, are called a electron transport chain. It's literally a chain of of these big complexes that accept electrons. So this is basically how it looks like. You have your NADH. This is all happening basically basically in your mitochondrial inner membrane. So around here, basically, these crystals. So in your membrane, your inner mitochondrial membrane, you have these big complexes. These are also electron carriers, like similar to NADH. We spoke about this before, that electron carriers can be actually floating around like an NADH. They can be in a membrane, and they can be actually swimming in the lipid tails, which we're going to be talking about. So what happens is that NADH is going to come in and it's going to pass its two electrons, and also it's going to basically remove its H plus. So it's going to pass its two electrons towards the first complex that is called NADH dehydrogenase. So the first electron carrier we have is called NADH dehydrogenase. It accepts these electrons and it will take this energy now, because this passing of this energy to here, we know that this electron is going from a higher energy level towards a lower energy level. That means that energy is lost. So what happens is that this NADH dehydrogenase, this complex, is going to use that energy. And this is actually other than just being an electron carrier, it is also a hydrogen pump. It will use this energy to actually do active transport of hydrogen from the mitochondrial matrix towards the intermembrane space. So this is where the hydrogens are. It's pushing them between here, in between the outer membrane and the inner membrane. That's the space we call intermembrane space. So this pump is able to take this energy and just basically pump them out, pump these hydrogens out. These electrons are now then passed to another basically electron carrier, which we've called ubiquinone, which we've labeled Q here. Ubiquinone basically is not a pump, it's just an electron carrier in the fatty acids. It's going to take him and it's going to pass him to another pump called basically BC1 complex. BC1 complex is an electron carrier, but also a electron carrier, basically. Sorry, a proton pump. So it's going to do the same thing as your NADH dehydrogenase. It's going to pump out these hydrogens. Then it's going to pass these electrons to cytochrome C. And cytochrome C is very similar to quinone. It doesn't do anything. It's just an electron carrier. It passes these electrons again towards the cytochrome oxidase complex. And this cytochrome oxidase complex also is basically a proton pump. So it's going to use this energy to push out hydrogens. Here, the electrons now has been passed on so many times that it's lost all its energy. It has barely anything anymore. So what we do is that now we pass it to a final electron acceptor. And that final acceptor is oxygen. And that creates water. It's oxygen when it's aerobic respiration. So remember, aerobic respiration means that the final electron acceptor is your oxygen. And then you give basically water out of that. So it's the passing of these electrons that you're literally giving energy to these pumps to push out hydrogens. Why would you need hydrogens to be pumped out? Well, because hydrogens, now what you've done is you've created a very big, let's say, kind of gradient in where the intermembrane space has much more hydrogens, so that it's much more positive compared to your mitochondrial matrix. So now these can go inside via passive diffusion. But the thing is that these are hydrogen, these are hydrogen ions. Ions do not like to go inside or in the middle of lipid membranes. They don't do that. They don't want to do that. So what happens is that these hydrogens, once they accumulate, they're gonna pass through an ion channel. And this ion channel is called ATP synthase, which we're gonna be talking about it. This ATP synthase is this very beautiful, nice structure in where you have this nice channel. So it literally it's like an ion channel. It's like this big channel that's embedded in the membrane, in your inner mitochondrial membrane. And then it has another basically unit here that looks literally like a kind of a doorknob. So the thing that you use to kind of open doors, it literally looks like that. So these multiple lobes all coming together in a circle. And what happens is that hydrogens are just going to pass through, and this is going to cause this head or this literally knob head to keep literally moving. And this movement produces energy that creates ATP. So this is why NADH can give us energy, because what you're doing is you're getting all these electron carriers and you're letting them pass these electrons, and this passing of electrons is fueling this active transport of hydrogens from your mitochondrial matrix towards your intermembrane space. So you're creating a very, let's say, high kind of concentration gradient in your intermembrane space, and you're allowing them this to go through passive transport. They're passing through ATP synthase. They're going through ATP synthase here basically now, and this is by now powering ATP. So this is NADH. FADH2 is extremely literally the similar thing, except NAD gives its first two electrons to NADH dehydrogenase. FADH2 doesn't. FADH2 gives it to quinone, ubiquinone straight away. It doesn't pass it to here. So NADH actually will cause more hydrogen protons to be pumped out because now you're literally making this NADH dehydrogenase work extra. So this is the difference between NADH and FADH2. So this whole thing of getting hydrogens, so you're creating a very big, as I said, high concentration gradient of hydrogen protons in your intermembrane space. The movement, the passive movement of hydrogen ions to go inside back into the mitochondrial matrix is called chemiosmosis. So it's literally the accumulation of protons in the intermembrane space, which drives protons into the matrix via diffusion. So as I said, all these protons are being cooked out basically by these pumps, and is fueled by the energy being passed around, by the electrons being passed around. So you create a big concentration gradient, like a much bigger amount of hydrogen protons in your intermembrane space, which then drives them to go back through passive diffusion. And this has been going in through this nice big channel called ATP synthase. And this literally causes basically this re-entering of ATP synthase. This causes the actual, basically, how can I put it, production of ATP. So chemiosmosis is the pushing or the movement of hydrogen ions into your basically mitochondrial matrix that gives off energy. That's chemiosmosis. So the electron transport system, it creates energy via chemiosmosis through the movement of hydrogen protons from the intermembrane space to your mitochondrial matrix via the ATP synthase. And that is the thing that creates energy. Does this make sense, or does anyone want me to repeat this? Okay. So, in a nutshell, this is basically all of aerobic respiration. You start off with literally glucose, which is then literally broken down into basically pyruvate. And then here it depends basically because it's aerobic, means that there's oxygen. Pyruvate is then taken up by your mitochondria, and then it's oxidized. So you remove a complete carbon dioxide. So this is a three carbon molecule. This becomes two carbon molecule. This then enters your citric cycle, which is further decarboxylated into literally nothing because acetyl CoA is just two carbons, and you remove two carbons here. But meanwhile, you're oxidizing it, you're releasing a lot of NADH and FADH2s. These then come inside and they need to basically pass on these electrons to your electron transport system. So basically, your NADH dehydrogenase will take the electrons from NADH. It will pass it to ubiquinone. Ubiquinone gives it to the BC1 complex, gives it to cytochrome C. Cytochrome C then gives it basically to this one, which now for some reason I forgot its basically name, which is basically cytochrome oxidase complex. So all these passing of these electrons basically will literally cause these pumps. So it's specifically NADH dehydrogenase, basically all these basically all these three pumps to push out hydrogens out from your inner membrane to basically so you're basically your inner compartment to your intermembrane compartment over here. And this is driving chemiosmosis. So this is literally aerobic respiration because at the end of the day, the electrons are being passed to oxygen, hence aerobic. Please do not get confused with that. So ATP synthase, as I said, is literally made of two different, let's say, kind of subportions. You have the F0, and this is your ion channel. This is the ion channel that is literally has this kind of channel is embedded in your membrane, into literally your inner membrane, what's it called? Your inner mitochondrial membrane, and that is allows the hydrogens to pass through. Then you have the F1, which is basically this knob, this whole head that literally looks like a doorknob. And this is what we call the F1, the catalytic head, because it's the one in where when the hydrogens are passing through, it's gonna keep turning around, and this is gonna catalyze the formation of ATP. So it's literally going to push the production of ATP. So basically you get ADP plus PI, so inorganic phosphate, and you phosphorate it. You use this energy to phosphorate this at the end of the day. So the F1 is the portion of the ATP synthase that does all of the catalysis, the basically, let's say, this kind of reaction. The F0 is more the ion channel. It's the thing that's allowing these hydrogens to pass through. So it's allowing chemiosmosis to occur. So now let's talk about the energy yield of respiration per se. So let's actually put this calculate this. So starting off with glucose, we said basically when you're literally breaking glucose to pyruvate, you actually get 4 ATPs, but you're literally giving 2 ATPs. Remember, this is basically you're kind of priming glucose to be broken down. Well, you're losing 2 ATPs, but you're creating four, so net you get two. Okay, this is great. and you get two NADHs, which is perfect. And then in aerobic respiration, pyruvate is oxidized. So when you do that, you get two NADHs, and then this is passing to your citric cycle. In your citric cycle, we get six NADH and two FADH2. Because remember, guys, we're creating two pyruvates. So this is doubled. Because we know that in pyruvate oxidation, you only get one NADH. Well, because there's two pyruvates, you get two NADHs. And we know that in your citric cycle, you get one ATP, you get three NADHs and one FADH. But again, because we have two pyruvates, it's doubled. Please, please do not get that confused. Here, everything is one, but starting from pyruvate, it's doubled because you get two pyruvates. So what happens is an NADH will then go into your electron transfer system. It's going to pass to your electrons, and they're going to create ATP through chemiosmosis. Great. One NADH can produce one and a half ATPs. Sorry, one NADH can produce two and a half. My bad. Two and a half ATPs. FADH produces one and a half. Because remember that if we go back in here, FADH2 will only pass electrons to ubiquinone, so it doesn't go here. NADH can produce more because it's pumping out much more hydrogens. So that's why there's a difference in number. So if you have a 2 NADH here, that means that you get 5 ATPs. So if you have basically 6, you're going to get 15, 2 FADH. So we know that 1 FADH produces one and a half, so 2 will give you 3. So if you calculate this all, you get a total net yield of 32 ATPs. So you get 32 ATPs from one glucose molecule. But this is only, you only find this in prokaryotes. In eukaryotes, you only have 30. You don't get really the full 32. Why is this so? Purely because the transportation of one NADH. So remember, if we go back in here, this NADH is that we create here. The transport of this to your mitochondria will cost you one ATP. And there's two that are being created. So you lose two. This only happens in eukaryotes because, as we know, prokaryotes do not have membrane-bound organelles. Some do, it's a rarity, but majority of them don't. It's only eukaryotes that have. We need to transport the NADH produced in the glucose that's in your basically cytoplasm. We need to transport it towards basically your mitochondria, and that will cost us one ATP. So this is why for eukaryotes, you get 30 ATP molecules. But for prokaryotes, you get 32 because there's no need for transport. You don't lose those two extra ATPs. So this is a very, really nice, let's say, mechanism, the whole literally, let's say, biochemical pathways that are connected to each other. But the thing is about it is that, as I once said, we don't want to deplete all our glucose. Once we have enough ATP, we need to stop because we have a storage of glucose, like glycogen. We don't want to run out of that because if you run out of glucose, you can die. Basically, you can actually go into hypoglycemia. It's called hypoglycemic shock, and you're basically kind of, yeah, you're going into seizures, and you start to actually lose consciousness. So we want to prevent this. So what happens is that ATP, too much ATP, it actually becomes a allosteric inhibitor towards phosphofructokinase. So you're stopping this reaction from occurring. So basically kinase, it allosterically inhibits ATP, but it also ATP is also able to stop citrate here. So ATP has this nice, let's say, function of inhibiting both the glycolysis but also the citric acid cycle. But also what we see is that NADH also is able to stop because, as I said, if you have too much NADH, it means that you have very little NAD because again, you have a pool of NAD and NADH. NAD gets transformed into NADH, and the more you produce NADH, the less NAD is basically being formed because it's just being used up. You don't want that. You don't want to come to the point where you don't have any more NADs. So what do? You basically is that when you have a lot of NADHs, this actually becomes an inhibitor to both pyruvate dehydrogenase, but basically will also inhibit in some sort of way, basically as well, fructokinase in some way. So ATP and NADH become, in some sort of way, inhibitors. They will stop this whole production when they're too much. So now let's talk about cellular respiration versus fermentation. So cellular respiration can be both aerobic and anaerobic. The difference between aerobic and anaerobic, aerobic, you use oxygen. Anaerobic, you use another inorganic, basically compound. This could be, uh, for example, methane. This could be sulfur. This could be other things other than basically oxygen. But both of them are quite similar. The only difference between them is what they're using. Aerobic uses oxygen as a final electron acceptor, and aerobic uses something else, another inorganic molecule. But both of them will keep reducing chemical compounds such as NADH and FADH2. They're going to keep breaking down glucose molecules to produce NADH and FADH2, and you're going to create literally a electrochemical gradient. So this proton gradient that we spoke about, this chemiosmosis. So in cellular respiration, you get energy from substrate level phosphorylation. So remember, this is in glycolysis. When you break down glycolysis, you're giving energy to phosphorylate ADP to create ATP, but you also use oxidative phosphorylation, which is technically chemiosmosis. Fermentation, no. Fermentation here, you utilize basically kind of, or these kind of organic compounds to accept these electrons, and you do not create an electrochemical gradient. By this point, you do not create NADH and FADH2s that plenty to create an electrochemical gradient. You don't. The NADH that you produced will give out its electrons straight into pyruvate to basically to be fermented. So in fermentation, you only take energy, you only create energy or ATP using substrate level phosphorylation. So this is the main difference between them. Cellular respiration will create energy from both substrate level phosphorylation, and then it's going to also get it from oxidative phosphorylation, which is technically chemiosmosis, because you're creating a electrochemical gradient, this proton gradient. Fermentation, it doesn't reach that point. It stops literally at glycolysis, and then basically you've done your ATPs through substrate level phosphorylation. Your NADH that you've created in glycolysis is passing its electrons straight away to pyruvate to create alcohol or lactic acid. So, an anaerobic respiration, as I told you, is using inorganic molecules other than O2. So that's anaerobic respiration as a final electron acceptor. It's literally the same thing. Anaerobic and aerobic are almost the same thing. The difference is what's that final electron acceptor? You could use sulfur, you could use nitrate, you could use basically carbon dioxide, and even you could even use our inorganic metals. Fermentation, you use organic molecules as a final electron acceptor. And usually that organic molecule is pyruvate. So this is the main difference. And here, because you're using these other inorganic molecules, you're creating a proton gradient for chemoosmosis. Here you're not in fermentation. So in a respiration, I'll give you a couple examples. You have certain basically microorganisms that are, we call them methanogens because rather than using your oxygen as your final electron acceptor, they use, for example, carbon dioxide, and it's basically, reducing it to methane. Hence why we call them methanogens, because they actually create a lot of methane from basically anaerobic respiration. And you see this a lot in cows. In cows, every time they're farting, they produce so much because they're using CO2 as their final electron acceptor. So they create a lot of methane. Why we have a lot of water as well with us is because we also create a lot of water during our respiration. Well, when it comes to cows, they produce a lot of methane. You have a lot of these kind of microorganisms, specifically certain archaea, that do sulfur. They use sulfur sulfate. So you use this kind of sulfur, or sorry, the sulfate, kind of an organic molecule, and this is reduced to hydrogen sulfide, this H2S. And this was shown to be like one of let's say, early stage for evolution for photosynthesis. This, we're going to come back to chapter 8. Don't worry about this sentence for now. But just for now, that you know that in sulfur prokaryotes, what they do is they will use sulfate, so SO4, to be reduced to hydrogen sulfide, technically. This is what they're going to do. And there's just an example of one of these kind of sulfurous kind of aspiring organisms. So you have this Thermoproteus tenax, which is an archaea species. So it's an archaea, technically microorganism. It lives in these kind of, let's say, kind of hot springs. And basically, this is one that you see in the Yellowstone National Park in the US. And they live in these areas that are so rich in sulfur. Because they live in these areas that are rich in sulfur, they've actually adapted themselves to use sulfur rather than oxygen as a final electron acceptor because their environment allows for that per se. Why we use most probably why us humans we use oxygen is purely because most probably we live in an environment that is rich in oxygen. This is why we're using oxygen. Well, these guys, they live in an area that's very rich in sulfur. So they're going to use sulfur as their source of a final electron acceptor. So fermentation. So as I said, fermentation, we're not creating a basically proton gradient. You're not doing that. You're not creating, basically, you're not going into citric cycle to create more FADH and FADH2s to go into electron transfer system. You're not doing that. So straight away from glycolysis, you create ATP using substrate level phosphorylation, and you have pyruvates. In yeast, so it's different between basically animal cells and yeast cells. In yeast, what happens is that first of all, pyruvate is decarboxylated, so you remove a carbon dioxide to create acetaldehyde, and then that is your electron acceptor. So as we said, in fermentation, it's organic compounds. So this is an organic compound. It accepts these electrons that we produced basically from NADH, and you get ethanol. This is how you create alcohol. As I said, you literally what you do with alcohol, no matter what type of alcohol, you take a sugar. You take a sugar from, I don't know, it could be from barley. It could be from, basically, what do you call it? Now I'm forgetting everything, basically. From fruits, from grapes that we create the wine. All these, you get those sugars. So all these glucose molecules. You literally put them, you entrap them in a closed container and you add yeast because yeast is going to use this glucose. And because there's no literally oxygen, because you close that container, what you're going to do is you're going to force it to go into fermentation because no oxygen means no aerobic respiration for the yeast. So you're pushing ethanol. So this is literally how you produce alcohol. Of course, when you do that, when you put this in a container, you need to put a small little opening because there's a lot of CO2 being produced. So it's a lot of gas. And if you do not have an opening, it can actually, the whole container can explode. In humans and animal cells, basically, we have a different thing where we straight away, we do not decarboxylized pyruvate. We go straight into pyruvate. We take pyruvate and we give the electrons to pyruvate to create lactate. And this is the lactic acid. This is the thing that makes our muscles very fatigued. So technically, we do also ferment pyruvate. We ferment pyruvate to create lactate. And this is basically what can cause us to get muscle cramps. One thing that we also see is that it's not only glucose that can be used as a source to create all of this kind of, let's say, energy. We can use proteins. We can use fats. We can use a lot of different stuff. Another example that I'll give you is, for example, we can do deamination. So there's a certain chemical reaction called deamination, and where you take glutamate. So this is glutamate. You deaminate it, meaning that you remove this literally nitrogen, what do you call it, group. You remove it completely, and basically glutamate now becomes alpha ketoglutarate. This can enter your citric acid cycle because if we go back to your acid citric cycle, so if we go here, it's here. So this can now enter and do this again, and you start creating NADHs, ATP, all of these guys. So proteins or amino acids can be actually used for to create ATP, not just glucose. Alanine can be converted to pyruvate. Aspartate can be converted to oxaloacetate. So oxaloacetate is again in the citric cycle. So you can create, it's not just glucose as the only molecule that can actually be used for respiration. No, that's not true. You could use also proteins. You could also use fats. So in fats, what you can do is, so these are your fatty acids, technically per se. You can actually do something called beta oxidation. In a sense, it's this following nice basically reaction. Again, I don't need you to remember this. This you don't need to remember. But what I want you to remember purely here is that beta oxidation uses basically you use ATP in a sense that where you're what you're trying to do is you're breaking fatty acids by two carbons because to create acetyl-CoA. So remember, acetyl-CoA is two carbon molecule. So what you do is you steal from the fatty acids, from the fatty acid tails specifically, you take from the fatty acid tails, you steal two carbons and you add a CoA enzyme and you have acetyl-CoA, which can then enter the citric cycle. So that's what you're literally doing. So in generally fatty acids, they are very, very, very big. They have a lot of carbons. They can go up to almost 60 different carbons. And usually most actually fatty acids have a not an a even number of carbons. So that's actually quite great. So if I tell you, for example, I have a six carbon fatty acid, how many acetyl-CoAs can you do? Three, because you divide it by two. If I have 10 carbon fatty acid, you get five. If I have 20, basically 20 carbon fatty acid, you get 10. So, which can then enter the citric acid cycle. And the cool thing about this is that if you compare a six carbon fatty acid with a six carbon glucose, we can get way much more energy from the carbon fatty acid. You get 20% much more energy because in the six carbon glucose, you create two pyruvates. In the six carbon fatty acid, you create three. So you get a 20% yield of an increase. So this is why fatty acids have much more energy than sugars. There are excellent energy sources for us, technically. And this is why burning fat will give you so much energy. And this is all because of beta oxidation. So again, I don't need you to remember this. Basically, how can I put it? The structures. What I want you to know is that what you do is that you take a fatty acid, you do a beta oxidation, basically will use one ATP and will generate basically from two carbons you generate an acetyl CoA and also FADH2 and NADH. That's all I want you to know. So this is what you get. So here you have basically, let's say the macromolecules that we studied about, nucleic acids, proteins, polysaccharides, lipids, and fats. We break it down into their literally monomers, be it nucleotides, amino acids, sugars, fatty acids. All of these can technically be broken or converted into basically creating pyruvate, creating acetyl CoA, going into your citric acid cycle. And then, of course, you have basically your metabolic products, depending usually what your final electron acceptor is. If it's oxygen, you get basically water. If you get NH3. So we can use all these different sources of these building blocks to create pyruvate or acetyl CoA or other citric acid cycle compounds. But one thing that we can also do is that we can actually use these products from the citric acid cycle or acetyl CoA to build. So remember, when you're destroying something, it's catabolism. So catabolism is when you're literally breaking down substances. So going from here to down here, that's called catabolism. But when we're trying to build something, when we're trying to build lipids or proteins, that is called anabolism. So we can use also certain molecules from the citric acid cycle, or we can use either also pyruvate or acetyl CoA to actually build up in basically macromolecules. So this is bidirectional, and it's a very beautiful, very nice metabolo kind of, let's say, loop that we have in ourselves, because there are times where we need energy, so we need to break down, but there's also times where our cells need to grow. So we need to stop a lot of this and we need to go into this area, more the anabolism, more the building. And it's very nicely, tightly regulated. So for my last thing, it's the evolution of metabolism. So one thing that we started to see is that we are very interested to see how do we actually evolve? How does our metabolism evolve specifically? So if we go really back in time to the most earliest creatures that basically we found, we started to see that none of these creatures did not have really the enzymes for glycolysis. They didn't have the enzymes for citric acid cycle. They didn't. They only had very simple kind of mechanisms and where they were able to actually store chemical energy and ATP. That was it. They were able to take any chemical energy around them and just create ATP. They did not have this ability to bring glucose molecules in to break it down or proteins to break them down to go into citric acid cycle. No, it was just purely they were managing to take just chemical energy, whatever they found, and put it in ATP. Later on, they started to actually evolve glycolysis. This was one of the most primitive, so glycolysis is one of the most primitive, let's say, reactions that we have. And it's a pathway that's found in all living organisms, going down from a plant to an ant to us. It's everywhere. And most probably it's stick to there because it's extremely efficient. It's so efficient in breaking glucose in a very natural way, even though it's 10 chemical reactions, it is able to actually extract as much energy as possible from it. And then later on, so basically, in glycolysis, you're breaking down or rearranging electrons or breaking chemical bonds, and you're using that breaking of these bonds to take up energy, right? Well, later on, basically, we started to see that certain plants didn't need to do that. They started to do basically photosynthesis, which is a bit different because in photosynthesis, you're using the energy coming from the sun. So you're not breaking technically you're not breaking chemical bonds. You're not doing that. You're just taking the energy from the sun, you're utilizing that to create glucose. And the really, really early ones, they did this anoxygenic photosynthesis, meaning that it does not produce oxygen. and they were mostly using basically this hydrogen sulfide. This might seem confusing, this anoxygenic photosynthesis and all these things, but don't worry, we're going to be talking about this in chapter 8, what this means per se. I don't want to confuse you already with these things, but just know that after glycolysis, we started to really start looking into photosynthesis. Photosynthesis was the next step, and specifically photosynthesis that doesn't use oxygen. Then this photosynthesis started to become basically use rather than using H2S, because photosynthesis uses water actually to drive the production of glucose. They started to use water, and this shifted everything because during this time where basically plants were not using basically H2S was purely because basically because there was not much water, there was not much sources of water, because during that time everything was very, let's say, dry. It was later on when we started to actually have a lot of water that basically we started to use water in our photosynthesis. And then a lot of permanent changes started to happen because when you break water, basically you're literally releasing oxygen. So during that time, our we started to people scientists started to see that our Earth started to be populated with much more oxygen. Again, these three and four, we're gonna dive into them, but just know that first thing we started to use energy to store an ATP, then went to glycolysis, and then from there we went into photosynthesis. It started off by literally, anaxygenic photosynthesis using H2S, and then we started to use water. Basically, then the fifth one is we started to use nitrogen fixation. Nitrogen fixation is that we are able to take nitrogen gas, which is N2. N2 is two nitrogens that are connected to three bonds. We were able to take them up and break those three bonds and use nitrogen, because if you look at nitrogen, nitrogen is a compound that is found in literally amino acids, in proteins, in basically nucleic acids. You need nitrogen. So our fifth, let's say, step was being able to use nitrogen, because during this time we were not able to actually really create so much proteins, amino acids. It's only when we started to be able to use nitrogen. And then the final and most evolved pathway is your aerobic respiration. This is your Krebs cycle. This is your electron transport system. This is how we think basically our metabolism started to change over time. Okay, guys. Um, this is the end of chapter 7. It's a very long one. I'm sorry for that and a lot of memorization. But again, if you guys need any help or anything, I've put so many videos where you can use everything, basically all these things. Please, please, please use them if you need any help. But also, if you need anything, please do let me know as well. Is there any questions, anything that I can help you guys with? Well, guys, if there's no questions, I hope you guys have a great continuation of your day. Stay safe, and please, 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 always feel free to reach out if there's something. Have a great one, guys. Thank you.
Thank you, doctor.
Thank you,
Thank you.
doctor.